Hello and a very warm welcome to everyone to our today's webinar on bioanalysis methods for respiratory drugs, achieving sensitivity, selectivity, and reproducibility. I am Vipul Chauhan and I represent Veeda Clinical Research Limited. Veeda Clinical Research Limited, Veeda together with its subsidiary Bioanalytical India Private Limited, together referred to as Veeda Group, offers a comprehensive portfolio of clinical, preclinical, and bioanalytical services to support innovative, biosimilar, and generic drug development programs of our global clientele. Together, we serve clients globally in pharmaceutical and biopharmaceutical, agrochemical and industrial chemicals, herbal and nutraceuticals, medical devices industries. As your facilitator today, uh, I would like to introduce you all to a couple of points as we begin this session. This session will have a presentation in the beginning, followed by a question and answer round. We have enabled our Ask a Question feature. It's right on your screen where you can drop all your questions related to this webinar right there. Now moving forward, I would like to introduce you to all our speaker and panelists for today's session. Mr. Samil Desai holds a postgraduate degree in quality assurance from the Postgraduate Department of Pharmaceutical Science. With around a decade of experience in bioanalysis, he specializes in utilizing sensitive methods on advanced equipment such as LC-MS, MS to meet both regulatory and sponsor demands. He has worked with notable organizations including Torrent Pharmaceutical Limited and Labna Therapeutic Research before joining Veeda Clinical Research Limited as an associate clinical manager in the Bioanalytical Research Department. Dr. Chirag Patel is working as a bioanalytical investigator at Veeda Clinical Research Limited, Ahmedabad, India. He has more than 18 years of professional experience in bioanalytical research. His role is to guide bioanalytical scientists in method development and method validation activities and supervise activities performed in regulated bioanalysis. He has published several research articles in peer-reviewed scientific international journals like the Journal of Chromatography B, Journal of Chromatographic Science, Journal of Pharmaceutical Analysis, Drug Testing and Analysis, etc. Mr. Jairaj Chudasma is working as a senior group leader at Veeda Clinical Research Private Limited, Ahmedabad, India. He has more than 18 years of experience in bioanalytical research. His role is to guide bioanalytical scientists in method development and method validation activities in regulated bioanalysis for generic products and new chemical entities. He has published several research articles in peer-reviewed scientific international journals like the Journal of Chromatography B, Journal of Chromatographic Science, Journal of Pharmaceutical Analysis, etc. Mr. Jairaj has also presented the scientific poster at different international forums like WRIB, EBF, etc. Dr. Vikas Trivedi is working as a bioanalytical investigator at Veeda Clinical Research Limited, Ahmedabad, India, where his role is to serve as scientific and technical lead for biopharmaceutical companies. Vikas has over 17 years of experience, professional experience, research experience of regulated bioanalysis in developing, validating, and applying bioanalytical methods to support clinical studies of generic products and new chemical entities. Now, without any further delay, I would like to request Mr. Samil to take over the session.
Thank you, Mr. Vipul, for nice introduction. Today, we will discuss about the following points. First is an introduction to bioanalysis of respiratory drugs, understanding sensitivity, selectivity, reproducibility in bioanalysis, current analytical techniques for bioanalysis of respiratory drugs, factors influencing method development and optimization, validation and regulatory compliance, case studies, future perspective and emerging technologies, and finally, conclusion. First, introduction to the bioanalysis of respiratory drugs. What is the importance of bioanalysis methods in respiratory drug development and clinical research? Bioanalysis methods play a pivotal role in respiratory drug development and clinical research due to several key reasons. First is the pharmacokinetic studies. Pharmacokinetic studies help to evaluate the quantification of drug levels in which bioanalysis methods help to measure the concentration of respiratory drugs and their metabolites in biological samples such as blood, plasma, serum, or axial breath condensate. Understanding drug absorption and distribution, also these methods aid in studying how respiratory drugs are absorbed, distributed, metabolized, and eliminated in the body, providing insights into their pharmacokinetics profile. Second is the efficacy and safety assessment. By quantifying drug concentrations over time, bioanalysis methods enable researchers to assess the effectiveness of respiratory drugs in achieving therapeutic outcomes. Also, by monitoring drug levels and metabolites helps identify potential adverse effects and assess the safety profile of respiratory medication. Third one is the biomarker identification and validation. Bioanalysis methods facilitate the identification and validation of biomarkers associated with respiratory diseases, providing valuable insights into disease progression and treatment purpose. Also, biomarker analysis allows for the development of personalized treatment strategies tailored to individual patient profiles, improving treatment efficacy and minimizing adverse reactions. Fourth one is the clinical trial support. During clinical trials, these bioanalytical methods are essential for assessing drug bioavailability, dose response relationship, and inter-patient variability, helping to inform dosing regimen and study protocols. Also, bioanalytical data generated through validated methods are crucial for regulatory submissions to agencies such as FDA, EMA, demonstrating the safety, efficacy, and quality of respiratory drugs. Fifth is the therapeutic drug monitoring. Bioanalysis method support therapeutic drug monitoring programs by monitoring drug concentrations in patients receiving respiratory medications, allowing clinicians to adjust doses regimen for optimal therapeutic outcomes. Also, therapeutic drug monitoring helps prevent drug toxicity by maintaining drug levels within therapeutic range while minimizing the risk of adverse effects. Six is the advancements in drug delivery systems. Bioanalysis methods aid in evaluating the performance and efficacy of various drug delivery systems for respiratory medication, including inhalers, nebulizers, and intranasal devices. Also, by quantifying drug deposition and absorption kinetics, these methods contribute to optimization of drug formulations to enhance patient compliance and treatment efficacy. In summary, bioanalysis methods are indispensable tools in respiratory drug development and clinical research, facilitating the evaluation of drug pharmacokinetics, efficacy, safety, and personalized treatment approaches. This application enables researchers and healthcare providers to make informed decisions regarding drug development, patient care, and the management of respiratory diseases. What is the challenges in analyzing respiratory drugs? Analyzing respiratory drugs presents several challenges that require specialized bioanalytical techniques and methodologies. Some of the key challenges include sample complexity, in which biological samples such as blood, plasma, serum, urine, sputum, and axial breath condensate contain a complex mixture of endogenous compound, proteins, lipids, and other components that can interfere with the drug detection and quantification. Also, matrix components can affect the performance of analytical instruments, leading to ion suppression or enhancement in mass spectrometric based methods. Second is the low concentration. Many respiratory drugs are administered at low doses, resulting in low concentration of drug and its metabolite in biological fluids. So also the bioanalytical methods must be highly sensitive to detect and quantify drugs accurately at these low concentrations, often required specialized instrumentation and sample preparation techniques. Third one is the metabolite identification. Respiratory drugs undergo extensive metabolism in the body, resulting in formation of numerous metabolites with varying pharmacological activities. So by identifying and characterizing these metabolites is crucial for understanding drug metabolism, efficacy, and safety profiles, but it presents challenges due to their diverse chemical structures and low abundance levels. Fourth one is the selectivity and specificity. Biological samples contain endogenous compounds that may interfere with the detection and quantification of respiratory drugs, leading to false positive or false negative results. Hence, analytical methods must exhibit high sensitivity and specificity to distinguish the drug of interest from structurally similar compounds and metabolites present in the sample matrix. Fifth is the sample preparation and stability. Sample preparation techniques such as extraction, purification, and derivatization are essential for isolating drug from complex matrices, enhancing analytical sensitivity. So, respiratory drugs and their metabolites must be prone to degradation during sample collection, storage, and analysis, necessitating careful handling and storage condition to ensure sample integrity. Sixth one is the method validation and regulatory compliance. Bioanalytical methods must adhere to stringent validation criteria outlined by regulatory agencies such as FDA, EMA, ICH to ensure the reliability, accuracy, and reproducibility of the results. So method validation involves assessing various parameters, including accuracy, precision, linearity, range, specificity, and robustness to demonstrate the analytical performance of the method. Addressing these challenges require multidisciplinary approach involving collaboration between analytical chemists and pharmacologists, toxicologists, and regulatory experts to develop and validate bioanalytical methods that meet the specific requirements of respiratory drug analysis. Innovative technologies such as mass spectrometry based techniques, high resolution chromatography, and advanced data processing algorithms continue to play a crucial role in overcoming these challenges and advancing the field of respiratory drug bioanalysis. Next is the understanding sensitivity, selectivity, and reproducibility in bioanalysis. What is the significance of sensitivity, selectivity, and reproducibility, and how these factors influence the accuracy and reliability of drug analysis? First is the sensitivity. Sensitivity refers to the ability of an analytical method to detect and quantify a target analyte at low concentration. High sensitivity is crucial for accurately measuring drugs and their metabolites, especially when they present at low levels in biological samples. It ensures that even trace amount of analyte can be reliably detected and quantified. Selectivity. Selectivity is the ability of analytical method to accurately measure the target analyte in presence of other interfering substance or analytes. It ensures that the measured signal is attributable to the target analyte, minimizing interference from endogenous compound, metabolites, or other coeluting substance. It enhances the accuracy and reliability of the drug analysis by reducing false positive or false negative results. Third one is the reproducibility. It refers to the consistency and reliability of analytical results obtained from repeated measurement of the same sample under the same condition. It ensures that the analytical method produces consistent results over time and across different operators, instruments, and laboratories. It is essential for establishing the reliability and robustness of the method and for ensuring the comparability of the results obtained from different studies or laboratories. How sensitivity, selectivity, and reliability influence on accuracy and reliability of drug analysis. Sensitivity enables the detection and quantification of drugs and their metabolites at low concentration, providing a more comprehensive understanding of drug pharmacokinetics and efficacy and safety profiles. It allows for the detection of subtle changes in drug concentration that may be clinically significant, particularly in therapeutic drug monitoring and pharmacokinetic studies. Improved sensitivity reduces the likelihood of false negative results, ensuring that even low levels of analyte are accurately detected and quantified. Selectivity minimize the interference from endogenous compounds, metabolites, metric components, or other coeluting substance that may lead to inaccurate measurement or false results. It enables the specificity of the method, ensuring that the measured signal is attributable to solely to the target analyte, thereby improving the accuracy and reliability of the drug analysis. Selective methods are less susceptible to the matrix effects and interference, resulting in more precise and accurate quantification of drugs and metabolites in complex biological samples. Third one is the reproducibility. It ensures the consistency and reliability of the analytical results, reducing variability and uncertainty in the drug analysis. It enables the comparison of results obtained from different samples, operators, instruments, and laboratories, facilitating data interpretation and validation. Reproducible methods are essential for establishing the robustness and validity of analytical procedures, ensuring the reliability of conclusions drawn from the drug analysis studies. In summary, sensitivity, selectivity, and reproducibility are critical parameters that significantly influence the accuracy, reliability, and validity of drug analysis methods. Ensuring optimal performance in these areas is essential for obtaining meaningful and interpretable results in pharmacokinetic studies, clinical research, therapeutic drug monitoring, and drug development. Third one, we will see the current analytical techniques for bioanalysis of respiratory drugs. The bioanalysis of respiratory drugs encompasses a variety of analytical techniques designed to quantify drug concentrations, identify metabolites, and assess pharmacokinetic parameters in biological samples. Here are some current bioanalytical techniques commonly used in the bioanalysis of respiratory drugs. First one is the liquid chromatography mass spectrometry, LCMS. LCMS is widely used for the quantification of respiratory drugs and their metabolites in biological fluids such as plasma, serum, urine, and sputum. Over its advantages: high sensitivity, selectivity, and specificity, allowing for the detection of drugs at low concentration and identification of multiple metabolites. Second is the gas chromatography mass spectrometry, means GCMS. GCMS separates the volatile and semi-volatile compounds using gas chromatography, followed by detection and quantification of mass using mass spectrometry. It is suitable for the analyzing volatile respiratory drugs and their metabolites, particularly those who have low molecular weights and high vapor pressures. Its advantages is its excellent separation efficiency, sensitivity, and specificity, making it ideal for pharmacokinetic studies and metabolite profiling. Third one is the biosensors. Biosensors detect the analytes by utilizing biological regimen such as enzymes, antibodies, or nucleic acids immobilized on a sensor surface. What is its application? So biosensors are increasingly used for the real-time monitoring of respiratory drugs and biomarkers in exhaled breath condensate or other biological samples. What is the advantages? It is rapid, sensitive, and selective detection, offering potential for point of care diagnosis and continuous monitoring in respiratory diseases. Fourth one is the enzyme-linked immunosorbent assay, ELISA. ELISA specific antibodies, it detects the specific antibodies and quantify target analyte based on antigen antibody reactions. It is suitable for quantifying respiratory drugs and biomarkers in biological fluids, including serum, plasma, and sputum. What is the advantages? It is simple, cost-effective, and amenable to high throughput screening, making it suitable for clinical research and therapeutic monitoring. Fifth one is the capillary electrophoresis. Capillary electrophoresis separates the charged analyte based on their electrophoretic mobility in a capillary field with an electrolyte buffer. It is used for the analyzing respiratory drugs and their metabolites, offering high resolution and short analysis time. Its advantages is it is excellent separation efficiency, minimal sample requirement, and compatibly with small volume of biological samples. In short, these analytical techniques play a crucial role in drug development, clinical research, and therapeutic drug monitoring of respiratory drugs, offering a range of options to suit different analytical needs and requirements. The choice of analytical techniques depends on the factors such as physicochemical properties of the drug, sample metrics, required sensitivity, specificity, and the stage of drug development or clinical applications. Next is the factor influencing method development and optimization. Wait. Factors. Previous slide, please. Method development and optimization in bioanalysis of respiratory drugs require careful consideration of various factors to ensure accurate, reliable, and reproducible results. Some of the key factors influencing method development and optimization include characteristics of drug and metabolites, the chemical structure of the drug and its metabolite influences the choice of analytical techniques, chromatographic conditions, and detection methods. Factors such as polarity, solubility, stability, volatility affect this sample preparation, chromatographic separation, and detection sensitivity. Second, sample matrix. Can you please forward to the previous slide? Sample matrix. Yes, sample matrix. Selection of appropriate biological matrix, such as blood, plasma, serum, urine, sputum, exhale, breath, condensate. Based on distribution and elimination characteristics of the drug, biological fluid can be selected. Second is the matrix effect. Understanding and mitigating matrix effects that may interfere with analyte detection and quantification, including ion suppression, enhancement in mass spectrometry based methods. Third one is the analytical instrument. Analytical instruments in which selection of suitable chromatographic systems like HPLC, GC, CE, based on physicochemical properties of the analyte and the separation requirements, and chromatographic techniques can be selected. Optimization of detection techniques such as MS, UV, fluorescence, ELSD to enhance the sensitivity, selectivity, and dynamic range of accurate quantification. Optimization of instrument parameters such as column, chemistry, mobile phase, composition, flow rates, temperature, and detector settings to achieve optimal separation and detection of analytes. Fourth one is the sample preparation techniques. Sample preparation technique selection of is important, in which extraction techniques like liquid-liquid extraction, solid phase extraction, protein precipitation, to isolate analyte from complex biological matrix. Also, implementation of cleanup procedure to remove interfering substance and matrix components that may affect chromatography, separation, and detection. Fifth one is the method validation. Adhering to regulatory guidelines, FDA, EMA, ICH, for method validation, including parameters such as accuracy, precision, linearity, range, specificity, and robustness. Also, development and execution of validation protocols to assess the analytical performance and reliability of the method under different conditions and sample matrices. Sixth one is the sensitivity and selectivity. Optimization of chromatographic and detection parameters to improve sensitivity and lower limits of detection and quantification. Also ensuring the selectivity. Implementation of selective sample preparation techniques and chromatographic condition to minimize interference from endogenous compounds and matrix components. 7 is the robustness and reproducibility. Evaluation of method robustness by assessing the impact of small variation in experimental conditions like pH, temperature, mobile phase composition on method performance. By conducting inter-laboratory and inter-operator studies to evaluate the reproducibility and transferability of method across different laboratories and analytes. 8.1 is the cost and time consideration. Optimization of method to minimize costs associated with reagent, consumable, and instrument maintenance without compromising analytical performance. Also, streamlining method development and optimization process to reduce turnaround time and increase productivity in drug analysis and research. So by carefully considering these factors and employing systematic method development and optimization strategies, analyst can develop robust, sensitive, selective bioanalytical methods for analysis of respiratory drugs, contributing to the advancement of drug development and clinical research in regulatory medicines. Next is the validation and regulatory compliance. Method validation is a crucial in bioanalysis of respiratory drug to ensure the reliability, accuracy, and reproducibility of analytical methods, adhering to regulatory guidelines set forth by the organizations such as FDA, EMA, and ICH is essential for obtaining regulatory approval and ensuring the quality and safety of the pharmaceutical products. Here is an overview of the importance of method validation, parameters involved, and compliance with the GLP and GMP. Importance of the method validation. First one is the regulatory compliance. Method validation ensures that the analytical procedure meet regulatory requirements and guidelines established by the regulatory agencies such as FDA, EMA, and ICH. Second importance is the data integrity and reliability. Validated methods provide accurate and reliable data, supporting decision-making processes in drug development, quality control, and clinical research. Third one is the risk mitigation. What is the importance? Validation identifies the potential sources of error, variability, and uncertainty in analytical methods, mitigating risks associated with inaccurate or unreliable results. Also, the inter-laboratory consistency. Validated methods enable consistent and comparable results across different laboratories, ensuring the reproducibility and transferability of analytical procedures. We will see next parameter of method validations. What parameter we can check by validation? First one is the accuracy, precision, specificity, linearity, stability of analyte in blood, matrix effect, robustness, range, long-term stabilities in matrix, also in solutions, short-term stabilities of solutions, stability of extract. Then, what is the compliance with the GLP and GMP? Compliance with the GLP and GMP requirements involves implementing standardized protocol, documentation practices, equipment calibration, personal training, and quality control measures to maintain the integrity and traceability of analytical data generated during method validation and routine analysis. In summary, method validation, according to the regulatory guideline, adherence to established parameter and compliance with the GLP and GMP standards are essential for establishing the reliability, accuracy, and reproducibility of bioanalytical methods used in analysis of respiratory drugs, contributing to the safety, efficacy of pharmaceutical products and clinical research outcomes. Next, we will see the case studies and applications. Here is some examples of successful bioanalysis methods for respiratory drugs. LCMS methods for the corticosteroid in axial breath condensate and bronchodilator in plasma. First one is the LCMS method, developed and validated for the quantification of corticosteroids like fluticasone in axial breath condensate. What is its significance? This method enable non-invasive monitoring of corticosteroid levels in patients with respiratory diseases such as asthma and COPD, providing valuable insights into treatment efficacy and adherence. Second is the LCMSMS method, which is developed to quantify bronchodilators such as salbutamol in plasma collected from the patient undergoing bronchodilator therapy. What is its significance? The method facilitated the pharmacokinetic studies and therapeutic drug monitoring, optimizing bronchodilator dosing regimen, and assessing patient response to treatment. Next, we will see the challenges. What is the challenges encountered and strategies employed? First is the what is the challenges is the sample matrix interference. Most of the bioanalytical methods is the challenge for the sample matrix interference. Biological samples such as sputum, exhale, breath condensate contain complex matrix that can interfere with the analyte detection and quantification. What is the strategy behind to remove these biological samples and clean the samples? So sample extraction techniques such as solid phase extraction and protein precipitation were employed to remove matrix components and enhance method selective and sensitive. Second challenge is the low analyte concentration. Respiratory drugs often administered at a low dose, resulting in low analyte concentration in biological fluids. So what is the strategy for detection? Enhanced detection techniques methods such as LCMSMS and highly sensitive detectors were utilized to accurately quantify drugs and metabolite even in trace levels. What is the application of these developed methods? These developed methods can be used, we have seen the pharmacokinetic studies, clinical trials, and in therapeutic drug monitoring. In summary, successful bioanalysis methods for respiratory drugs have addressed challenges related to the sample complexity, low analyte concentration, and matrix interference through innovative analytical techniques and robust validation protocols. This method have been applied in pharmacokinetic studies, clinical trials, and therapeutic drug monitoring, contributing to the advancement of respiratory medicines and improved patient care. Next, we will see the future perspective and emerging technologies. Potential advancement in bioanalysis techniques for respiratory drugs. First one technique is the microsampling techniques. Advancement in microsampling technologies allow for the collection of smaller volume of biological samples, reducing sample requirements and enabling minimally invasive sampling methods for respiratory drug analysis. Second is the miniaturized analytical platforms. Development of miniaturized analytical platforms, including microfluidic devices and lab-on-chip systems, offer rapid and high-throughput analysis of respiratory drugs and biomarkers with reduced sample and reagent consumption. Third one is the advancement in mass spectrometry. Improved mass spectrometry techniques such as high-resolution mass spectrometry and tandem mass spectrometry enhance the stability, selective, sensitive, and speed of drug analysis in respiratory samples, enabling comprehensive metabolite profiling and identification. Fourth one is the emerging sensor technologies. Integration of novel sensor technologies, including wearable biosensors and implantable devices, enables real-time monitoring of respiratory drugs level and physicochemical parameters in patients, facilitating the personalized treatment strategies and remote patient monitoring. Next is the integration of novel technologies. What is the integration of novel technologies like microfluidics and lab on a chip system, nanotechnology based sensors, mass spectrometric imaging? These methods is integration in novel technologies. What is the importance of continuous innovation? By continued innovation, addressing evolving challenges, enhancing patient care, and accelerating drug development. In conclusion, the integration of novel technologies such as microfluidics, nanotechnology, and mass spectrometric imaging, coupled with continuous innovation in bioanalysis techniques, holds great promise for advancing respiratory drug analysis and enhancing patient care in the field of respiratory medicines. Finally, conclusion. In conclusion, the continuous refinement and innovation of bioanalysis methods for respiratory drugs have revolutionized drug development and therapeutic monitoring in respiratory medicine by achieving heightened sensitivity, selectivity, and reproducibility. This advancement paved the way for improved drug efficacy, safety, and patient outcomes in management of respiratory diseases. Finally, what is the Veeda expertise? Veeda has developed and validated 1,227 methods till date, and among these, 100 methods are of new chemical entity. Veeda has developed methods for endogenous molecules, hormones, steroid, inhalation formulations, chiral and liposomal products. Average processing capacity of 1 lakh sample per month. And Veeda worked as a central bioanalytical laboratory for global phase 2 and phase 3 trials. Thank you. It's time for the question and answer.
Now moving on, we will be proceeding ahead with our discussion where we are going to answer some of the interesting questions posted by our audience. So my first question to Dr. Chirag is, why sensitivity is questionable for formulations like inhalations?
Yeah, thank you, Vipul. So for inhalation products, the drug dose is directly delivered to target site. So formulation dose is very low for these inhalation products, resulting in ultra low level drug concentration in body, like picogram or sub-picogram. This leads to the requirement of 1 picogram or lower level LLQ requirement, and establishing this LLOQ from the complex matrix like human plasma, where thousands of other components are also present in the plasma, becomes very tricky and challenging. And due to this, high sensitive equipment is required to achieve this sensitivity for these inhalation products method. Thank you, Vipul.
Uh, Thank you, Dr. Chirag. My next question to Mr. Jairaj is, what precautions to be taken while optimization of method for respiratory formulations?
Yeah, thank you, Vipul, for providing me an opportunity to answer these questions. Actually, optimization of method for respiratory formulation is always challenging in terms of sensitivity and cross contamination, and hence this require very careful attention, specifically at low levels, which is typically at picogram and sub-picogram levels. So, in this method optimization, if someone has selected solid phase extraction, then it is very essential to select the appropriate sorbent beds to eliminate the interference from the matrix samples. Further, for example, you can use this ion exchange or any suitable sorbent based on the molecule chemistry. Further, this kind of molecules always require a special care during sample processing activity to avoid the cross contamination, and hence it is always suggest to handle the sample at isolated place. So overall, I can say that from sample preparation to sample acquisition, each and every step has to be monitored very carefully for the successful execution of bioanalysis of respiratory formulation. Thank you, Vipul.
Thank you, Mr. Jairaj. And my last question to Dr. Vikas is advantages of using LCMS equipment from other analytical equipment for respiratory drugs.
Yes, thank you, Vipul. The first advantage I can say as compared to the other analytical technique which is available like ELISA and electrophoresis, GCMS. So the first main advantage for usage of the LCMS is the superior specificity, selectivity, and the sensitivity. Because as compared to traditional immunoassay, LCMS enables the accurate results, reproduce, and the false positive and negative results. Second point is speed and high throughput capabilities. Like for LCMS method meets the analytical need and budget limitation with the throughput, speed, and sample capacity to boost the workflow productivity for the laboratory development test where the analyte resolution is the critical. Third point is comprehensive and flexible, like simultaneously detection of multiple analytes in the single run. So we can run the more than 2, 3, 4 analyte in the single run, and flexible option to meet the sensitivity throughout our need. And fourth advantage is cost saving and increase the productivity because the fixed cost of the test decrease when the more tests are run in a single run, as well as the ultra fast selection reaction happens. So thank you, people. Over to you.
Thank you, Dr. Vikas. Lastly, I, on behalf of Veeda Clinical Research, would like to sincerely thank you all for being here today and taking out the time to be a part of this webinar. I would also like to extend our gratitude to the speaker for being such a wonderful presenter and giving us all great learnings today. We at Veeda Clinical Research Limited honestly look forward to hosting you all again. Till then, take care and stay safe, everyone. Thank you.